Thank you, Miguel. Thank you very much. And congratulations for the beautiful work, a nice paper, and also congratulations for your award. For the sake of time, we're going to move to the next session, but as I say in the previous session, please, if you want to make any questions to Miguel or some of the other speakers, you can do it through the chat that you have in your platform. If we have time, we will get a few of those questions at the end of the sessions. Otherwise, I will encourage you to use the platform for the posters, where you can also get into a discussion directly with the authors in there. Thank you very much, Miguel. So as I say, we're going to move to the next session, which is what we call the flash talks, or oral communication selected from the posters that we have received. And without much delay, I would like to present the first of the speakers. Will be Itziar de Rojas from Boada's group in the Fundació ACE Barcelona. Itziar, please go ahead.
Hi all, thanks Miguel for introducing me. My name is Itziar de Rojas from Fundació ACE Barcelona, and I'm here to talk about the common variants in Alzheimer's disease. The novel association of 6 genetic variants with Alzheimer's disease and risk stratification by polygenic risk scores. From now on, I will refer to Alzheimer's disease as AD. There are over 50 million people living with dementia, and the global cost of dementia is well above $1 trillion. This means there is a medical and economical urgency to efficiently test interventions that are under development. As AD is the most common neurodegenerative disorder, AD has become the next medical challenge of the 21st century. Genetic discoveries are the drivers of our understanding, and polygenic risk stratification can contribute towards planning of feasible and efficient preventative and curative clinical trials. The most prominent locus, APOE, was detected almost 30 years ago using linkage techniques. In addition, genome-wide association studies of AD case-control datasets and by proxy AD case-control studies have identified 30 genomic loci that modify the risk of AD. These signals account for approximately 31% of the genetic variants of AD, leaving most of the genetic risk as yet uncharacterized. Methods. We first perform a meta-GWAS integrating 3 sets of summary statistics using fixed effect inverse variance weighted meta-analysis. We combined a Spanish case control study called Durante de GESCO, the case control study of the International Genomics of Alzheimer's Project, IGAP, and the UK Biobank AD by proxy case control study. The genome research at Fundació ACE, we call GRACE, is a de GESCO project, which means Dementia Genetics Spanish Consortium. The GESCO is made up of various centers spread throughout Spain, as seen in the map of the right. More than 50,000 samples were collected from the GESCO centers, and genotyping was conducted using the Axiom Spanish Biobank array. After study-specific variance filtering and quality controls procedures, we have more than 12,000 Spanish samples available. In brief, the IGAP summary statistics. In brief, the IGAP is a large genome-wide association study using individuals of European ancestry. Stage 1 comprised samples from 4 consortia: HGC, EADI, CHARGE, and GERARD consortium. In stage 2, 11,000 of SNPs were genotyped in an independent set of case control samples. Finally, the meta-analysis of stage 1 and stage 2 comprised more than 82,000 of individuals. Details on data generation analysis have been previously described in Kankel et al. The UK Biobank. For this study, we used the published summary statistic of Marioni et al. They include unrelated individuals for whom AD information was available on at least one parent in the UK Biobank. In brief, the participants whose mothers had dementia were compared with participants whose mother did not have dementia. The same has done for the participants with and without paternal cases. The phenotype of the parents is independent, and therefore the estimates could be meta-analyzed. After an analysis, the estimates were made comparable to a case control setting. To check the estimates, we perform a sensitivity analysis, removing the 80 by study, and compare the results effect estimates with and without this dataset. After meta-analysis, we test all variants with suggestive association, p-value less than 10 times -5, located in proximity of genomic regions selected for follow-up. Next, we confirm the novel observed association in 16 independent European ancestry cohorts, many of them collected and analyzed by the European Alzheimer's Disease Biobank, EADB project. Then, we construct a polygenic risk score, PRS, based on the genome-wide significant variants showed in the present study, excluding ApoE to check the impact of PRS modulating ApoE risk. PRS uses common variants with a small effect and combine them into a one larger to predict dementia risk. The PRS was generated by multiplying the genotype dosage of each risk allele for each variant by its respective weight and then summing across all variants. We weighted the variants by the effect size from previously studies, Kunkel, Sims, and Jun et al., in a clinical AD cohort and in a pathological AD dataset. We tested whether the effects of the PRS were influenced by diagnosis certainty, sex, and age at onset groups in Grafe de Gesco study. Lastly, we test whether the PRS could be used to identify individuals at the higher risk of AD, and we compared age at onset of the AD cases. Results. Using this strategy, we identified a genome-wide significant association for 36 independent genetic variants in 34 genomic regions. The pink signals are the novel ones, in blue the previously reported, and in green the ones that required confirmation. Using our meta-GWAS approach and follow-up analysis, we identified novel genome-wide significant association of 6 genetic variants in AD risk, which are in PRKD3 and DUFAP7, PLC-gamma2, CHRNE, and APP genes, and 2 missense variants in sharping gene. We also confirmed a stopped codon mutation in the interleukin-34 gene, increasing the risk of AD, and 2 other variants in PLC-gamma2 and HS3SD1 regions. Although we were not able to replicate two loci in chromosome 14 and 15 that show suggestive association with AD in the discovery. Also, in a sensitivity analysis, we found a higher correlation between the effect estimates from the case control and by proxy approach for the significant loci. The most interesting finding from our GWAS is the discovery of a common protective intronic variant in the APP gene. Our results directly support APP production or processing as a causal pathway not only in familial AD but in common sporadic AD. The SNP is a DNase hypersensitive area possibly involved in the transcriptional regulation of the APP gene. This SNP is an EQTL for the APP mRNA and an antisense transcript of the APP gene. Functional evidence supports a modified APP transcription increased the TFCP2 transcription factor ability to its binding site and increased the enhancer activity of this specific intronic region. Based on this evidence, we can postulate that a lifelong slightly higher APP gene expression protects the brain from AD insults. Still, this seems counterintuitive, as duplication of the gene leads to early onset AD. A U-shaped effect or Hormesis effect of APP might help explain our observations. An alternative hypothesis is that mechanisms underlying the variants are related to the overexpression of protective fragments of the APP protein. Disentangling the molecular mechanism of our findings will help to refine and steer the amyloid hypothesis. Additionally, 2 independent missense mutations in sharpin increase the AD risk. These variants identified are altering protein sequence or affecting regulatory motifs. Sharpin was previously proposed as an AD candidate gene, and its component of the LUBAC complex that plays a regulatory role in inflammation. The variant located in the ChRNA, which encodes a subunit of the cholinergic receptor, is a strong modulator of ChRNA expression. The same allele that increases AD risk increases the expression in the brain and other tissues, according to CTEXT. We construct a weighted polygenic risk score, and we show that the association of the PRS with clinically diagnosed AD cases is similar to the association with pathologically confirmed AD. In these settings, adding variants below the genome-wide significant threshold did not lead to a more significant association of the PRS with AD. Next, we investigated the effects of the sex and age at onset. Our analysis revealed that the effects of the PRS was the same in both sexes and was consistent with both early onset and late onset AD. PRSs has the potential to identify subjects at risk of complex diseases. The PRS was associated with a gradual risk increase when we stratified the dataset into 2% percentiles of the PRS and with AD within the APOE genotype groups. We compared the risk extremes and found a 16-fold increased risk for the highest PRS group, APOE 4-4, compared with the lowest PRS group, APOE 2-2-3. Lastly, we studied the effects on age at onset of the polygenic risk score in APOE genotype groups. We saw that the polygenic risk score differentiated age at onset only within APOE 4 carriers. And when we compared the age at onset in 80 patients in the extreme risk groups, we found a 9 years difference in the median age, a 4 years median onset difference within APOE 4 heterozygotes, and a 5.5 years median age onset difference within APOE 4 homozygotes. Altogether, we described 6 novel loci associated with sporadic AD, among which one common APP variant. These signals reinforce that AD is a complex disease in which amyloid processing and immune response plays a key role. We add to the growing body of evidence that the polygenic scores of all genetic loci to date, in combination with APOE genotypes, are a robust tool that are associated with AD and its age at onset. These properties make the polygenic risk score promising in selecting individuals at risk to apply preventative therapeutic strategies. Especially, I would like to thank Agustin Ruiz from Fundació ACE and Sven van der Lee from Amsterdam UMC for helping me with this work, the GRAFE team, all partners, all collaborators. And thanks for your attention.
Thank you.